So first, we would like to introduce the project management team. So all of us here on the call. Mark, if you would like to give it a start.
Hello, sorry. I can never find the unmute button when I'm in a hurry. Hi, everyone. I'm Mark Spalding. I am a senior scientist in the Nature Conservancy. You'll probably see the light getting dark outside as the call goes on because I'm in Italy. And I've always I've worked on large scale ecosystem mapping and more focusing on ecosystem service mapping as well for quite a long time now. And so super interested in particularly in the kind of building the big picture, and our arguments for building the big picture has always been about building from the bottom up, using the field data that people generate to tell much bigger stories and hopefully to influence policy. So that's me.
We continue with the TNC team. Emily is on the call.
Hi, yes, good to be on this call and see so many participants. I am Emily Landis. I lead the Global Ocean and Climate Strategy at the Nature Conservancy. We have been extremely interested in seagrass for decades upon decades and been really excited to hear about the carbon value and the carbon interest in seagrass systems. And so this is just a very timely meeting for us. So thank you, everyone. And now let's move it to Lindsay.
Hi, everyone. I'm Lindsay. I am an ocean and climate scientist on the Global Tackle Climate Change here at the Nature Conservancy. My role here at TNC is understanding coastal ecosystems and how they function as natural climate solutions. So that involves mangroves, tidal marshes, and seagrasses. And I do that, or I quantify these roles using a combination of remote sensing, geospatial modeling, and quantitative social science methods. I think Dr. Steve Schill is unable to join today, as is Valerie McNulty, but Dr. Denise Perez is on the call, and I think she can introduce herself next. Denise?
Yeah, thanks, Lindsay. So I'm a spatial ecologist with the Nature Conservancy based in Puerto Rico. A bit about my background is I worked for quite a few years in seagrass and coral reef ecology in the Pacific and now in the Caribbean. But I also have a background in remote sensing, so more interdisciplinary, doing habitat mapping with satellite imagery. Yep, so I'll pass it off to FIU team to continue with the introductions.
All right, since no one's unmuting, I will. Hi, all. I'm Jim Forkran. I'm the PI funded by Nature Conservancy with a bunch of co-PIs on this project. I'm a seagrass ecologist, a biogeochemist, and I've become very interested in the blue carbon world. And I'll talk more about the project and the genesis for it after we get done with introductions. And Dr. Campbell, you're next.
Thanks, Jim. Hi, everyone. My name is Justin Campbell. I'm also a seagrass ecologist and assistant professor here at FIU and part of the CariCAS team. I'm also the founder and lead PI of another seagrass network, the Thalassia Experimental Network, which is a separate network of 13 sites that were established across the Caribbean and the larger Gulf of Mexico back in 2018. So I have some experience with sort of coalition building and also working with international partners in some remote locations. And I'm excited that a lot of the sites from my former seagrass network are joining us here with CariCAS. So I'm looking forward to sort of reconnecting with old colleagues and also meeting all the new members. So thank you. I'll pass it on to, I don't know, who wants to go next from the TNC, perhaps Juan? I'm sorry, from the FIU team.
I can do it.
Yep.
Okay, I am Juan Gonzalez. I've been doing marine ecosystems monitoring for about 10 years. And for the last year, I've been working in the Seagrass lab with Dr. Forkurian like in charge of the field operations and leading the Seagrass monitoring programs we have here at FIU. I think Johannes can continue.
Hey, I'm Johannes Krause, and I'm a postdoc in Jim Fochran's lab. I'm interested in seagrass ecology and seascape ecology, and so I combine a little bit of remote sensing with underwater ecological methods. Also interested in sediment biogeochemistry, so this project nicely spans my fields of interest. So I believe that Tom Frankovich can't make it today. Is Tanya on the call?
Don't think she can make it either today.
Okay. In that case, I think that wraps up our introductions. So next we will just have a brief high-level overview of the project itself.
Yep. Thank you, Johannes. And this is going to be very brief because we're going to be talking much more in depth about what we're doing and why we want to do it later on. But in general, we've come together to try to reduce the uncertainty about our knowledge of carbon stores and seagrasses in the Caribbean. And part of that is reducing our uncertainty in the distribution of seagrasses within the Caribbean. So we're trying to combine within this project some intensive in-water work and laboratory work, but also trying to do everything in a way that is relatable back to remote sensing and to the remote sensing expertise that TNC brings to the project. We're really interested in the idea of trying to use common techniques and a common framework across an entire region. This basically is 80% plus of the range of the seagrass species that occur in the Caribbean are represented by the range of sites we have from Panama and Colombia up to Bermuda and in the north of Mexico. So we've got a great group of people that say they all want to work together. And so we're building this network, and it would be a shame if this network just came together and wrote one paper and then went away. I'm really hoping that we're building a durable scaffold that will hold up much more collaborative work across the region. We want this to be a durable network. And I want to call out Emmett Duffy, who's on the line. Emmett started showing us in the seagrass world what could be done with the Zoster experimental network, Zen. And then Zen gave rise to 10, the Thalassia experimental network. And now we're just trying to do Zen and 10, but bigger on a regional scale. And we're learning, we stand on the shoulders of giants and the people that have done this before us. So we want to build this durable platform, this durable partnership. And we also want everyone on the call to be thinking about them and their graduate students and their own projects. And if there are things that you would love to be able to do regionally, I'm hoping that you can find a group of people to work with within this network so that we can do side projects hanging on the same, hopefully field work, but hanging on the same scaffold. So great to see everybody here. We've got 42 on the call now. I think that really exceeded all of our expectations. So Lindsay, are you guys, you want to talk about how a bit about the remote sensing that we already have done and how we want to tie the field work to remote sensing?
I was going to turn it over to our lead on our team, Emily, to talk a bit about the high-level overview of the project, and then we can jump into some of the remote sensing pieces.
Yeah, thanks, Lindsay. Just quickly also, I think it's important to just set the context as well from that global perspective. Right now, blue carbon is a very hot topic. Nature Conservancy is actually leading a blue carbon market project in Virginia, looking at eelgrass restoration to generate carbon credits. Countries are increasingly submitting their nationally determined contributions to the Paris Agreement and including things like seagrass systems. Yet for us, the system still needs to be explored far more than it has been. And I've definitely talked with Jim about this many, many times of not understanding, well, what really is the carbon content of seagrass in the Caribbean, because it's so informative for all around the world to really understand the value of seagrass. And so we're both not overselling seagrass, but we're also not underselling seagrass. And so the data that you're working on and the data that you're collecting is foundational and fundamental to telling that global story of what seagrass can do. So just thank you, because this is the science that I need. This is the science that policymakers need. This is the science that private companies need when they're thinking about carbon credits. So just thank you for what you do. It's really important. And Lindsay, I'll hand it over to you then.
Should we go to the next slide or?
Surely. Yeah, thank you for this overview. I think then it would be time to jump into the specific background of what we want to do in terms of blue carbon work and also remote sensing work. I think, Jim, do you want to jump in over here? And unmute. Jim, I think you're muted.
Pardon me, while I'm embarrassed about speaking and saying really important things while muted. Yeah, this is our dress rehearsal for these workshops, and we hadn't actually been through the script before, so we're jumping around a little bit. That's, I think, a little bit why we're kind of slow on the take up. I think it's my job now to just talk a little, to put blue carbon in context. I think this figure should not be foreign to anybody. In my lifetime, there's basically been a 100+ parts per million increase in the average concentration of CO2 in the atmosphere. And that is having all kinds of consequences in terms of global temperature, the distribution of water, the distribution of heat in the ocean, the amount of CO2 in the ocean, the pH of the ocean and the rain. So this is an anthropogenically generated problem, and we're all looking for ways that we can surmount this issue. So I can't change them, right? So blue carbon has an interesting history of, I'm going to give you a very quick thumbnail of it. The idea that most of the CO2 that goes in the atmosphere comes from fossil fuels is very well established, but then there's a component of the CO2 that's leading to that rise that is not coming directly from the burning of fossil fuels. It's from the conversion of land and land use from places that were carbon dense and stored a lot of carbon to places that have much less carbon in them. And when that carbon is lost to the atmosphere, that increases, it accounts for about 20% of the increases of CO2 in the atmosphere. And one of the most obvious ways that we're putting CO2 in the atmosphere from land use and land cover change is chopping down of forests and turning that wood into CO2. And that was so obvious that long ago, the United Nations started working on programs like RED, the reduction of emissions through deforestation and degradation. And RED sought to decrease that land use, land cover change component of the increase of CO2 in the atmosphere by helping people choose ways to use their land in ways that didn't lead to huge CO2 increases in the atmosphere. It didn't put CO2 in the atmosphere. So RED became a very popular program internationally because it provided money for landowners to not convert their forests into cattle pasture or shrimp farms or whatever else is being converted to, and receive compensation from the international community for doing that in the term of carbon credits. So RED is very well established, and it's running around the world, and it's doing a lot of good, not just for CO2 in the atmosphere, but also for conservation of land. And so it's an added on benefit. And in the early 2000s, mid-early 2000s, people were looking at pictures of the Earth and said, you know, red's great, but almost the whole planet's blue. Isn't there must be some value to the carbon that's in the ocean that could be used the same way as this red carbon is on land. And so beginning in 2005, 2006 or so, people started looking at this and wondering if we could develop policies like RED around the conservation of ocean carbon and keeping that ocean carbon from being changed into the atmosphere. So some places, there's a real clear relationship between RED program and blue carbon. For instance, in this mangrove forest, it's in Costa Rica. I'm standing about 10 feet off the ground, and that tree that I'm looking at is at least twice my diameter, 15 feet off the ground. So there are some forests that have a really, really high carbon content that are not usually managed by forest services, those mangrove forests. They're usually managed around the world by departments of fisheries because they're in the ocean, they're below mean low water. So blue carbon really hit the ground running when we just pointed out that these are just forests. And not only do they have lots of trees, but they also have a tremendous amount of organic carbon stored in anoxic soils beneath them. So go to the next slide. And Dan Donato and others published the first estimates comparing those things that we think are think of as so important in climate change negotiations and mitigation strategies, keeping tropical upland forests, temperate forests, and boreal forests as forests because they're so carbon dense. Then when you look, though, just at this first blue carbon habitat, mangroves, there's a tremendous amount of carbon that's stored in mangroves, and it makes them much more carbon dense than most terrestrial forests. And the stuff that's in the light gray there is the stuff that's in the biomass. And then the darker gray is all that carbon that's stored in soil. And if that soil is at risk to be released to the atmosphere, if we cut down the trees, then that soil is also actionable under carbon policy. So then, of course, blue carbon, as a brand, if you will, really focused on the vegetated coastal habitats for a number of reasons that I won't go into now, but many of them are policy, but they're also really, really carbon dense environments that are being lost quickly. So it did a brief summary of what we knew about carbon in seagrasses back in 2012. And you can see that there's a whole bunch of organic carbon stored in seagrasses, but you can't see that light gray because there's very little organic carbon stored in the biomass. It's almost all in the soils. And that array of points means that there are some seagrass meadows. In fact, that one is the top one there is a Posidonia meadow in the Mediterranean that have just as much or more carbon in them as any of these other important carbon stores that are policy actionable. So seagrasses have a lot of carbon, but the carbon is in the soil. Let's go to the next one. So how much carbon is there? Well, there's a whole bunch of uncertainty. I think some people say there's 172,000 square kilometers of seagrass in the world. And that's based on the minimum number or the minimum area mapped as polygons in the Seagrass Atlas from quite a few years ago. We think that there's probably at least 300,000. This is people like Carlos Duarte sitting around guessing. And there could be up to 600,000 square kilometers of seagrasses in the world. But that's a huge amount of uncertainty for anything that we want to do, that we want to use for policy and for practical reasons. So we hope that this project helped us get a much better idea of how much seagrass there is in the Caribbean. Just as a side story, there are 18,000 square kilometers in South Florida that aren't even included in that 172,000. So we know there's a lot out there. So there's about 2.5 megagrams of carbon biomass per hectare and about 140 megagrams of organic carbon in soils. And that means there's about 1 billion tons of carbon stored in seagrasses around the world, but that's come from using these numbers that are known with great uncertainty and multiplying them by a number with even more uncertainty. And we really want to try to narrow that down. Let's go to the next one, please. I'm still stuck on that one. Are you guys seeing the next slide? All right. So seagrasses are being lost at a rate greater than the loss rate of forests. And if we take those uncertain numbers that I showed you on the last slide and assume that if we lose 1.5% of them per year and the carbon in the top meter will eventually make it back to the atmosphere, there's a loss of seagrass biomass on the order of 15 teragrams carbon per year and a loss of seagrass soil organic matter on the order of 150 teragrams carbon per year. And if you add those up, it's potential that the loss of seagrasses could be driving about 10% of the total CO2 fluxes attributed to land use land cover change. So there's a potential for this being an incredibly important flux, but there's still a lot of unknowns. And we're working in this project to try to help reduce the uncertainty in aerial extent and reduce the uncertainty in how much carbon there's stored, and tie things that we can measure from remote sensing with things that are important for carbon to see if we can then use remote sensing to help make a much more accurate estimate of all of the organic carbon in seagrasses of the Caribbean. I think that's the last one I had. Yeah, so now after that, hey, Johannes,
Yep.
Open up and tell me, do you want me to talk about the author agreement, or are we going to do something else now?
Let's continue with the introductions and jump into the author agreement.
Excellent.
With the
all right, thank you guys.
Topical introduction, and after that, sure. So yeah, thanks, Jim. I was in this context about to talk about the data that we plan to collect with the CariCAS network. So at each of the network sites, as Jim said, we plan to characterize the soil carbon stores and also the seagrass growth at 2 meadows. So the idea is that we would like to measure seagrass abundance in the seagrass meadows alongside with abundance of other benthic organisms such as macroalgae and sponges. We are interested in the seagrass species composition, above and below ground biomass, and the seagrass shoot density. Now, looking at the soils, we plan to measure the organic and inorganic carbon concentrations from sediment cores, as well as nutrient content, and that would be nitrogen and phosphorus for us. We want to collect seagrass tissues and analyze them at Florida International University for their nutrient content, again, carbon, nitrogen, and phosphorus. And we also plan to measure stable isotopes of carbon and nitrogen on the soil, as well as the seagrass tissue samples. So let us look a little bit at the remote sensing aspect of it, which will build up on these field collected data.
Thank you, Johannes, for reviewing the data that's going to be collected at the field sites. I think that provides a really nice foundation to discuss how we can leverage this really rich field data collection effort in hopes to scale with remote sensing and geospatial modeling. So currently, a lot of estimates of seagrass blue carbon across broad extents are derived from point measurements that are scaled by total meadow area to provide estimates of total stocks, which is great, but there's quite a bit of spatial and temporal variability that really can greatly influence the accuracy of these scaled estimates. We're hoping to use these robust field estimates in combination with high resolution satellite imagery and geospatial modeling to scale blue carbon estimates in meaningful and more accurate ways. We hope to develop methods that will leverage this field data and the satellite remote sensing data and environmental proxies thought to drive variability in soil carbon to map the spatial variability of carbon at regional scales. And so leveraging this great field data collection effort and existing data that is already available, we have three approaches or plans, and one of which is to explore the feasibility of developing more detailed mapping of seagrass environmental characteristics, so seagrass density, cover information, potentially species information, and ultimately we hope to get at above ground biomass. Secondly, we hope to develop a spatially explicit model of soil organic carbon using a combination of that great field data and environmental covariates thought to drive that variability in carbon storage. Finally, we can leverage this field data, presence and absence information, species information, to refine existing seagrass habitat maps that are available for the Caribbean. And so now I'm going to hand it over to Denise to discuss the Caribbean team's work in developing seagrass extent maps for the region.
Great, thanks, Lindsay, for passing over. So just to start, in order to be able to even get maps of carbon and blue carbon, which is the ultimate goal, you really have to understand how can we map that accurately and be able to understand what the actual extent is for seagrass, because there is a lot of uncertainty. And so with remote sensing, there's different types of products that we can use, and we're planning to use satellite imagery. But depending on the area that you're looking in and how much of a budget you have, is going to depend on really how good that data is going to be and that how good the imagery is going to be. And so here is an example of different spatial resolutions where, you know, the 1/2 meter resolution is very expensive, and it's like the highest resolution that you can really get for satellite imagery. And there you can really be able to detect and pick out where seagrass occurs. And in this example, you can do it pretty accurately just because that this species grows very densely and has really long seagrass blades. But if you're looking at a species that grows very sparse or has really short seagrass blades, even looking over the side of the boat, it's going to be really difficult to detect unless you're in the water. And so that's why having field data that hopefully will be collected this summer is going to help so much with being able to validate and provide the calibration that we need for mapping seagrass more accurately. And we're really excited about this project because we have access to 1/2 meter resolution imagery through our partnership with Planet, which has a constellation of satellites. And so we're hoping to use that imagery to be able to test out methods for mapping out seagrass in more detail, like species and density, as Lindsay mentioned before, and maybe even have better accuracy in being able to separate algae and seagrass, because since they have very similar photosynthetic pigments, it's always a challenge and it's always really confusing to be able to map that apart with satellite imagery. And in contrast with something that's more medium resolution, like around 3 meters, which is a lot of what multispectral satellites come as, you can see the loss of information that you get because it's more pixelated, and you're not going to be able to get the well-defined patches of seagrass that you can detect with higher resolution. And then with other satellites that have coarser resolution that are like 20 meters or even higher, that are typically free and are really great for regional or global analysis, there's even further loss of information. So there's always going to be a compromise depending on the size of the area that you're trying to map. Maybe you can go to the next slide. Thanks. So for the current seagrass maps that we have, we're working with one that is medium resolution, which is the Planet Dove satellite imagery. That's about 3.7 meter resolution. This was published in 2020, and this is the first time that we were able to get higher resolution imagery across the whole Caribbean. And this took over 2 years to be able to collect, because in the tropics, it's really cloudy. And for satellite imagery to work, you really need cloud-free, good imagery. So it took 2 years to be able to collect all of these scenes and mosaic it together to create this regional map. So the final product that we came out with, if you go to the next slide, is the seagrass extent maps for the insular Caribbean. And this was extensively validated using underwater video, as well as consultation with local experts. And these maps are available online right now. So you can visit this website. I'll copy the link into the chat, and you can explore for your country, or if you want to do regional analysis, it's all freely available, these layers to download. And there's also even a link if you are an expert and you know maybe there's an update that's needed where maybe we didn't have it mapped out, the seagrass, or maybe there's an area that we mapped as seagrass, but it's really something else, you can go to the link on this website and be able to give us back feedback as well to update. And for us, this isn't already published, finalized map, but we want to keep on updating it as well. And so with the field data that's going to be collected through this partnership, we're hoping to use that data to help refine and update these maps as well. And you can go to the next slide. Okay. So Lindsay, I think you wanted to take over on this one.
Thanks, Denise. And I apologize, Johannes, it's a bit of an animation, so you may have to, yeah, thank you. Even though seagrass above ground biomass represents a small short-term carbon pool, it has the potential to influence carbon stocks, particularly in the soils. And recent studies have demonstrated there is a relationship between soil organic carbon and seagrass biomass. So we're hoping to leverage those existing extent maps that Denise was just talking about, which are already divided broadly into like dense and sparse classes, in combination with existing field data and the data that will be collected as part of this project. And we're hoping to explore the feasibility of using remote sense data to add information on species information, cover classes, and ultimately above ground biomass. And as we said, the largest carbon pool within these seagrass systems is within their soils. And because they can't be measured, soils can't really be measured directly with optical satellite imagery due to the imagery's inability to penetrate the water column, we really must rely on these like environmental proxies, many of which though are derived from remote sensing, to estimate the carbon stores in the soils. And the hope is to map soil organic carbon in the study area, we can identify important covariates or proxy indicators through a combination of lit review, expert opinion during workshops such as these, and follow-up focus groups or interviews, as well as the preliminary results from the field data collection to gather or develop spatial data related to these proxy indicators. And then we can use these as well as our other outputs from seagrass cover and potentially above ground biomass for use in spatial regression models to hopefully predict in a spatially explicit way soil organic carbon across the study area, using that field data again for training, testing, and independent validation efforts. And so if you want to go to the next slide, we put, through an initial consultation with experts and from existing reviews that are out there already, we've pulled a list of potential drivers of carbon in seagrass soils. And we know, this is not an exhaustive list, and not all of these proxies are available as spatially explicit data sets. We aren't necessarily limiting our analysis to existing data sets, but it's an important consideration in addition to understanding what these environmental proxies are. Do we have spatial data sets in existence for them currently? And this list is just to get us thinking. We hope to follow up with you in focus groups or in one-on-one interviews, but if something stands out as important to you that you don't see on this list right now, feel free to just add it in the Zoom chat, or if you feel really strongly about a certain characteristic here or driver as one of the main ones or the must-haves, feel free to put those thoughts or comments in the chat as well while we move on. And so now I will pass it along to the next speaker. Or if Mark, did you want to?
If I can just say two quick things. The first, I wanted to just let people know that we've done this before. With other habitats. And so there's a mangrove soil map that was published a few years ago, and we've just finished another one which will be published very soon, where it's exactly the same approach, field data plus this environmental proxies to build a global model. So it's very doable. That was the first thing I wanted to say. So we're quite excited. And the second thing, and this may come up, and sorry if I'm preempting it, but everyone on this call is invited to join in this process as well as the field work. I think we're really keen that this is all of us contributing to building the bigger picture. So it would be great to have everyone involved. As Lindsay's saying, if you have ideas, send them in. If you have thoughts, send them in. If you have other data, send it in too.
Thank you, Mark.
Lindsay, this is Jim. I have my hand up, just addressing a question that came up in the chat. the question is,
Mhm.
why didn't TNC focus on any of the mainland adjacent seagrasses in that mapping effort? And we, on the other hand, in this group, we've put quite a lot of emphasis in getting those mainland sites to add data as well. And it's my hope is that those data will then go in to inform the mapping effort that allows TNC to expand to the mainland adjacent seagrasses as well.
That's a great question. And I'll start to answer it, and then I may pass it over to Denise. But so you are correct in that the Caribbean team in their mapping efforts focused on the insular Caribbean alone. And I think that's a result of their focal area as a team. And so as part of this project, you are correct, Jim, in that we are hoping to expand in ways to the degree possible. And we are hoping for those areas that don't necessarily have existing maps, we're hoping to reach out to the folks on the call, partners, to understand if they are aware of local to regional scale mapped efforts to have in concert with some of these field data collection efforts. I'm not sure if that answers your question, but Denise can also provide, if there's anything that I left out, Denise can provide additional context probably on the extent of the mapping in the Caribbean.
Yeah, Lindsay, and Jim, thanks for answering that. Yeah, you're both correct. Our team focuses on the insular Caribbean, and so that's where a lot of the field collection happened. And that's why it ended up being that only including those countries that are currently through the Caribbean ringmaps.tnc.org on that website. So.
Thank you. If that answers the question, we would move on. So I would like to present the anticipated project outputs that we hope to generate through this effort here. And some of them were already briefly mentioned, but the general idea is that this field data collection and then the integration with remote sensing products would lead to 2 synthesis papers. One focused on the seagrass carbon stocks and environmental drivers across the Caribbean, and a second one building on what Lindsay presented in terms of spatially explicit modeling of seagrass carbon stocks. Then, of course, Jim touched on it briefly, but we hope that this network that we generate here would also lead to other undertakings and projects that might span the region, maybe just a few subsets of sites or perhaps just one site. But we hope that the bringing together this great collection of researchers of the region will enable some more site-specific papers that collaborators might want to work on. Other than those publications that we are planning, there is, of course, going to be a mapping outcome of this. So we hope to have seagrass carbon stocks maps and create a map from this. There's also going to be a project database. So all the field data that we collect is going to be centrally stored in a database that is going to be available first to all the project partners, and then also publicly open source available. So we hope to integrate the data that is collected through CariCAS with existing repositories like OBIS and the Coastal Carbon Atlas, so that these data really reach wide and benefit the wider research community. Furthermore, for the field and laboratory methods, we developed a field guide, which comes in booklet form. And this will also be available not only to every partner in this network, but also to the public. And lastly, of course, we hope that this is going to be a lasting network of Caribbean seagrass scientists who can collaborate in the coming years, even when the official project timeline ended. So I think this might be a good moment to go over our draft authorship agreement. Jim, if you want to share, I stop sharing this for a moment. if you could unmute yourself and
So you all just see now the authorship guide in a Word window, right?
yep.
Okay, good. So we're working under a general guiding principle of inclusivity rather than exclusivity. We want credit to go where credit is due for people that work on this large project, and to try to put some bounds on that and to make sure everybody understands, we have a written authorship guide that we're going to ask everybody that becomes a partner to sign on to. It's too long to read, but I will say that the number one guiding principle is be inclusive. The number two is we will start off as we're writing papers with everybody who contributed data or was important at a site's name on the paper, and then those people individually will have to opt into the paper and say, yes, I think that I've done enough to be included as an author. We have a list of criteria that everyone will be responsible for themselves going through and being honest with themselves if they have met these criteria, and if they have, they deserve to be authors on the paper. So that's for the largest, the two big papers that Johannes talked about. For any other smaller projects that come out of this work that were inspired by the work, we ask that you reach out to the lead team of CariCAS and offer authorship as appropriate to the lead team as well, since that was part of the reason that the paper came together and the data were collected and the data were analyzed. Now, we'll send this out to everyone and ask everyone to sign it. I will give you an example of how this works. So for the Thalassia Experimental Network, we just got one paper accepted for publication. It has 44 authors on it from 13 sites. And I will tell you that the review process on that paper went so incredibly smoothly after having to deal with 44 different sets of reviews before it even actually went out for review. So it's more work for the authors, but it makes a better product. So we want everyone to benefit from the academic products of this network, and each person is going to make the decision for themselves whether they've done enough to be included. Justin, you want to talk more about the way this authorship agreement worked in 10, or did I cover it well enough?
Hey, Jim, no, I think you covered all the ground. So as Jim mentioned, we just want the network to be inclusive in regards to including people who were important in collecting data and addressing questions across the network. So as Jim mentioned, I mean, the network agreement, the authorship agreement is roughly 2 pages, and that'll be sent out to everybody for you to review and then hopefully sign and then send back to us just so that we make sure that everyone is on the same page.
Great, thank you, Jim and Justin. So our next point on the agenda would be the project materials and resources. So we would like to talk a little bit about what the project brings to partner sites and what the partner sites bring to the project. I think that would be you, Jim.
Unmute. So we really see this as a partnership. We can't afford to do everything, and we can't afford to provide everything, and you may need things from us. So it will be our role as the central project to provide sets of key field and laboratory equipment for temporary use at the partner sites. We've put together 10 big field boxes that include the cores that you'll see later and the vials that will be used and all of that stuff. We hope that, and then here in the yellow, we'll give you a steel core, the the piston for that core, because we're going to use a piston core to collect uncompressed cores. We'll send you a biomass core, an herbarium press, and herbarium paper, because another thing, a side project that I have interested in, maybe someone here is better to run that side project, is the taxonomy of seagrasses in the Caribbean is a mess. And it would be really nice if we could collect voucher specimens of all species from all sites so that we can get DNA and morphology data from those. So herbarium supplies will be coming with you. We'll send you mesh bags and these 20 milliliter vials and subcores and a field methods guide. We'll ask you to return the equipment and or send it along to the next person that needs it, next lab that needs it, and then send the samples back to us. And here at FIU, we're going to run inorganic and organic carbon content, nitrogen content, phosphorus content, and carbon and nitrogen stable isotopes. We're use those carbon and nitrogen stable isotopes to try to look at the amount of that organic matter in the soils that's actually being contributed by the seagrasses. We'll provide a site for the database, and we will publish the data in repositories, and we will be lead on synthesis papers. And we can also send visiting field personnel if your site needs them. So the next point is what we need from you. We're going to rely on you to attain permits, because you guys know your countries way better than we do, for field collection and for export. I will send specific instructions about export permit labeling or export labeling when you mail seagrass sediment samples back to me. It's important that we all use the word sediments instead of soils, because marine sediments are not regulated, soils are. So we'll rely on you to obtain permits. We'll rely on you to provide field personnel and boat time and access to the site or wherever you're going to get to it. And, some minor field and laboratory materials. It would be, we hope that you all have access to a drying oven, to an analytical balance, to GPS, your own waterproof paper and a printer. Things that we need to collect the samples, like aluminum foil, rulers, razors, pencils, a metal rod 2 meters deep, underwater measuring tape, a compass, a camera, clipboards, quadrats, and a tripod, a winch. We may still include the winch in our kit. You'll see pictures of that later. Towels, knife, duct tape, etc. So we've got a kit of things that we think you're probably not going to have. We've made some assumptions about what you probably are going to have. We're going to need you to be the field laborer and ask us when you need help with it. And we'll be the lab labor, and we'll all work together. Johannes, I think it'd be better, instead of taking questions now, because I'm sure like a lot of people have questions now, let's wait until we see the methods in action, and then we can talk about what individuals need and what wasn't clear from this. All right.
Yeah, that sounds like a great idea. We're going to have an extensive section about the intricacies of each field method. And I think this will give you additional background to judge whether you do have the specific materials needed or the ability to take those samples. But before we jump into the field methods, let us briefly touch on the site selection. So in this case, because there was a question in the chat, this refers to the selection of seagrass meadows to be sampled at your location. Justin, do you want to cover this one?
Sure, I'll chat quickly about site selection. So what we're looking for is for all of our partners to select 2 seagrass meadows within their location. And we have a primary site and sort of like a secondary site. The primary site should be a seagrass meadow that perhaps you might be more familiar with, one that you have perhaps a personal interest in, a site that perhaps you have historical data from, perhaps a location where you've done prior experiments. That's a site that we would like the partners to select based upon their own sorts of interests. And then we would like partners to converse with us about the selection of the second site. And the partners you all should think about perhaps 2 or 3 other alternative sites that are selected in consultation with us. And so these could be sites that are at a slightly different depth, at a slightly different exposure, at a slightly different seagrass density. And so we'll have essentially 2 sites that all partners will select. One will be our primary site of their choosing, and then the secondary site will be selected in consultation with both the FIU and the TNC team. So this is sort of obviously the initial workshop, and then there will be a process whereby we will follow up with partners individually to help sort of figure out exactly what that second sort of alternative site will be. And I believe, Johannes, you mentioned that there will be some sort of Google form application that we will send out to our partners where they can fill in some basic information. Is that correct?
Yeah, that is right. I'm not sure, Lindsay, maybe you wanted to talk to that as well, at least relating to the remote sensing aspects.
Sure. Yeah, I think just in terms of a scaling opportunity and from a remote sensing perspective, when we thought about, we talked about those environmental proxies, I think it'll be interesting and important to try to capture some of that variability across our sites and locations. And so when you're thinking about those alternative site selections, we were thinking it would be great for you to think about, if you jump to the next slide, Johannes, do you have seagrass meadows across this continuum of, low to high density, shallow, deeper water, different habitat geomorphologies. So yeah, when you're coming up with those alternate sites, it'd be really helpful from a remote sensing and scaling perspective to capture some of that variability, if at all possible. And as Justin and Johannes said, we will work with you to identify the most efficient and effective secondary site.
Thank you, Lindsay. Yeah, so as Jim suggested, we will defer the questions to the end of the next section, and then we go through the questions in chat and whatever other questions you might have besides that. But now I think it would be time to talk a little bit more in depth about the field and laboratory methods that we propose to be executed at each of the partner sites. Juan, did you want to start on this?
Okay, of course.
Or should I start on this?
However, okay, this is our the most tricky part that we are asking for you to do. That is the field methods. It's really not too tricky, but we have to follow protocols, and this is we can divide the field part of the sampling in 2 parts: the corings, the sediment coring, and the seagrass abundance and biomass. Next. So here's the site setup. We need a metal rod to locate our site. We have to make sure to get the GPS coordinates as accurate as possible, so we suggest we take multiple readings and then average them. And we can also use a buoy to mark the site. This next one, please. This metal rod is going to work as the central point of our sampling site. We need a hammer to dig the rod, and I think that's it for this part. Next.
Sure. So next, I'm gonna show you the sediment core tube preparation. And just to repeat, we are going to send these specific core tubes to all the partners. And they're not your typical off-the-shelf tubes. These are stainless steel tubes that have sampling ports or holes along the side of them, so that we can extract subsamples at specific depths after we took the sediment core directly and analyze these. And so we don't have to transfer the material out of the core barrel, extrude or anything like that. So I'm going to show you again a little video clip of what that looks like. So this is the core barrel with those sampling holes. And before we take the core, we have to cover those holes. So we use duct tape to bring it along the side of the core. So for the duct tape to really stick, we wash it first with fresh water and then dry it off well with a towel. So when it's really clean and dry, then the duct tape sticks best. It's important to have the duct tape really snug fit on it, because if there are wrinkles or so, it might rip off when you drive the core in. And then we wrap duct tape also around the bottom to stabilize, and just above the top hole of the core. So when we drive the core into the soil, we see this duct tape that is wrapped around just above the top hole. And that signals us we have to stop driving it into the sediment, because we can only really sample from the top hole, and that should be at the sediment surface. So here in the video, you see us wrapping this duct tape just above the top hole. And then we work with a plunger on a metal chain that is inserted inside the sediment core tube. These plungers, they can be adjusted in width so that they just snugly fit inside the tube. And the idea is that this is going to cap the core and create suction if you remove the core out of the sediment, so everything stays in place. It's going to prevent the compaction of the core as well. So once the core tube is prepared, it pretty much looks like this. It has all the subsampling holes covered with the duct tape, and the plunger is inside, metal chain sticks out the top, and then it's basically ready to be used. The plunger, the metal chain as well, is something that we will provide and send to you when you agree to take part of this network. So the materials that are needed for the core tube preparation are, of course, the core tube itself, a little bit of fresh water and a dry towel, the duct tape, plunger, and metal. So basically all of this is going to be provided to you. So what we found that works really well is to use multiple layers of duct tape, one just centrally across all the holes, and then another 2 layers, 50% offset to the sides. Again, if we are not careful to lay the duct tape really closely to the core barrel, but if we create little bubbles or wrinkles or so, that really gives opportunity to the sediment to catch on it. And then if you drive the core into the sediment, it might just rip off the duct tape, and then you have to start over again. So really care should be taken to make sure this is really smooth. I think I covered the rest here. So once the sediment core tube is prepared, we want to set up our coring site. And our site marker that we installed before, this metal rod driven into the sediment, that's the position that we measure really closely with the GPS. And right next to it, we want to take the sediment core to be sure that it relates, the sediment data that we get from it relates to the site location. I'll pass it over to Juan.
Okay, so to set up the station, to help the coring, we attach a metal chain to a tripod or to a metal ladder. This tripod can be built anywhere. It's a super easy tripod to build. We used that wooden board just to help us to put the tools we are using in the coring, to put them in the board. You can use it if you need it. That board is just a piece of wood with weights on the corners, and we put those wood sticks to place the tripod there. For this part, you just need the prepared core. We need to use a metal rod to make sure that we have enough sediment, because we need the cores we want to analyze, we want them at least, if we can, a meter depth. So we have to test with the metal rod, hammering the rod into the sediment until refusal. It means until you cannot dig it anymore, to check if we have enough sediment to take the sample. We must make sure that the core is totally vertical and perpendicular to the board. Okay, to pass to Johannes.
All right. So after the core station is set up, of course, we have to take the sediment core. So now that the core is sitting there upright, the metal chain is attached to the tripod, we use a hammer to just drive the core into the sediment until it's not possible anymore to drive it further. And our maximum depth, again, is this duct tape that we wrapped above the top hole of the core. So even if we could drive it further, we really don't want to do that, because everything that's all the sediment that would sit atop the top hole, we can't subsample. So we really want to stop there. After we reach that point, we secure the metal chain to the core here with a vice grip, so that the plunger stays in place when we retrieve the core. Then we attach a little rope on the side. This is a timber hitch that we, but you can use any knot that works for you. And then we attach a winch to the tripod and the rope, and can easily extract the core here without too much manpower, just using the winch. And of course, once the core is retrieved, we have to make sure to really quickly cap the bottom so we don't lose any sediment if it's unconsolidated. And after that, transport the core, in this case, to the boat, if you're coring your shore, to the shore. But it's important to keep it upright during transport. So we don't want to shake it too much and make the sediments move inside. So we really want to just hold it upright after we extracted it. So for this task, we just need the coring station that we set up before, a hammer, the rope, a vice grip, winch, and a core cap. Again, the core caps, we're going to send you probably also the winch as well. So really rope and hammer are what you would have to provide here for this task. And again, it's just important whilst you drive the core into the sediment to make sure that it's really staying vertical and you don't come in at an angle, because we want the depth measurements to be precise. And if you drive it at an angle, yeah, we're losing a little bit of that precision there. Okay, do you want to talk about subsampling the core, Juan?
Yes, sure. Once on the surface, we just peel back the duct tape to find the sediment surface. And an important part in this step is to attach a tape to measure the top of the sediment core. And then we proceed, we continue taking small subcores of the core tube. these subsamples, we take them with these cut up syringes with a previous known volume. We have to write down the depth, the sample volume, and the vial label, the vial number. We should repeat that subsampling until you reach the bottom of the core of the metal tube. For this section, the materials you need are the metric tape, the tape to measure, the cut-up syringes that we are going to provide you, the pre-weighed vials, and a knife or a spatula to clean up the surface of the sample. You have to make sure that once you write down the volume, once you have the volume in the syringe, you have to take care of putting everything, all what you have in the syringe, all the volume in the vials. For this part, next, Johannes, please.
Well, I'm sorry, Juan, before you go on to this next part, may I go back one slide and talk about a couple of things?
Please.
So this is Jim again. So I don't know if it was obvious to you watching this, but what really we're trying to do is create two different sizes of piston cores. So a piston core is a device that's made to be pushed into soil or sediments with suction so that you don't get compression of the core. So that's the reason that plunger is on the end of the chain. And that in concept is exactly the same as these cut-off syringes. So the syringe is cut so that it's square on one end, and you put the plunger of the syringe at 0 volume. And then if you saw in the video, the plunger body is pushed in while, the body of the syringe is pushed in while the plunger is held constant. So the whole idea is we're trying to collect samples without compression, because we need to have a dry bulk density. We need to know how much material there is per volume if we're going to be able to get carbon calculations done correctly. So that big stainless tube with all the holes in it is just like a cut-off syringe, just bigger and longer. So that's one point I wanted to make. And then another point I wanted to make is not all seagrasses have a meter plus soil or sediment underneath them. The reason you're using that probing rod to begin with is to find out how much you have. And if you only have 40 centimeters and that's your site, you can core at 40 centimeters. But we won't expect the core to go any deeper than that, so you won't drive the core into the basalt. All right. So when it bottoms out, it bottoms out. But what that means then is, as you're pulling the tape off of the core to expose the holes, the first 60 centimeters of holes are going to just be filled with water. And then you'll get down to the plunger, and then you slow down because right under the plunger is the 0 depth subsample. So we're sampling with depth relative to the top of the soil column. I hope that was clear. If not, well, we can do questions later. All right, sorry, Juan.
Okay, thank you, Jim. Before going to the field, we should have pre-weighed vials with no caps. And then you can label them, label the caps, just for you to know which vial is which, to keep track of those. And after the sediment collection, we need the wet weight after the collection, and then the dried weight. The dried weight, we take them until we get a constant weight, but if you leave the sample from one day to another, that should be enough to weight the samples. Remember the vials, we have to put them in the oven uncapped. So for this part, what we need from you to provide is a drying oven and a scale, analytic scale to pre-weight and then weight the vials.
One we may also, depending upon capabilities, we could pre-weigh vials and send them pre-weighed. I've had a long discussion with Anna about that, so I think we are prepared to do that for everybody that needs it.
Yes, we can provide the pre-weighed vials, and in that case, you should send us the wet samples. If that's the case, if you don't have access to an analytic scale, we can just give you the pre-weighed vials, but we would ask you to send us the wet samples to have to do the weighing here at FIU. Here is the I don't see it quite well, but yeah, here it is. Here we have like the data sheet we're supposed to use in the lab and in the field, and these are the rows you should help us help us fill out. That is nothing else than the vial ID, the vial initial weight, uncapped, the vial plus the sample wet weight, then the vial plus the dried sample, and the total weight of the sediment. And we also collected, you are also going to collect the volume in the field, and then with this data we can get the dry bulk density. Next.
All right. So I think we switch gears here a little bit because this is basically the sediment sampling done, creating the sediment core, taking it and analyzing the data. Again, at FIU, that's where we're going to measure the inorganic and organic carbon as well as nutrients and stable isotopes. So once you send off the samples, that is what's going to happen on our end in the lab over here. Now, to relate this soil carbon data to seagrass growth and to something that is remotely sensible, can be remotely sensed, we want to collect seagrass above ground and below ground biomass, as well as abundance and so on. So you've seen in the beginning that we are using a central stake to mark the site. And this is going to be our center point for finding quadrat locations at which we are serving seagrass abundance and taking biomass cores. So we would like to have a total of 16 quadrats relating to each seagrass meadow. And these should be placed in a somewhat randomized location. So the way that we do this is we are generating 16 radial coordinates. That means 16 compass directions from 0 to 360 degrees and distances from 1 to 30 meters that combined make up radial coordinates. So we have an example over here in this table that you see on the bottom left, where each quadrat has an angle and a distance that combines to a direction and distance to swim. So what we do to make sure that these quadrats are somewhat evenly distributed around the center stake and not all just clustered, for example, in one direction, is that we want to make sure that the angles that we generate, that 4 of them are between 0 and 90 degrees, then another 4 between 90 and 180, another 4 between 180 and 270, and then the other 4 between 270 and 360. So on the right side, you see a schematic drawing of this. So essentially, we have these randomized quadrant locations, 16 of them, but 4 for each quadrant. And this will hopefully allow us to have robust ground truth data for our remote sensing aspects, as well as to collect data that is representative of the immediate surrounding of the coring location, so that we can make inferences about sediment carbon based on seagrass characteristics. Now, we don't want to collect 16 biomass cores. That would be a lot of biomass cores. We are thinking of collecting biomass cores in every second quadrant. So again, looking at this table, there's a column for biomass core. It just says yes or no. We would like to collect 8 biomass cores in total. And we can do this just right after we score seagrass abundance in each quadrant, or depending on how we plan our dives, we might also just stick a surveyor's flag in every second quadrant location and come back to those locations later to collect all the biomass cores. Say if we want to first survey seagrass abundance in the 16 quadrants and then do the 8 biomass cores, then we would use these flags to make sure that we are collecting the cores from the quadrant sites. Again, alternatively, we can just, after scoring a quadrant, collect the core right in the quadrant and then move on to the next place and score. So this is the general idea of the site setup. Juan is going to talk a little bit about what we do at each quadrant location specifically.
Yes, okay. So once we reach every quadrant, we measure the canopy height. You can bring a ruler. For example, we use the clipboards we have here. They are measured, so you can use whatever tool works best for you. We are going to determine the sediment texture, the sediment type, and we are going to score the abundance of seagrasses, all species you find, of macroalgae, sponges, and corals. The steps to do this is first navigate to each quadrant location, then take a picture of the clipboard, of the paper, for you to know which quadrant are you sampling. Then take a picture of the quadrant. For that then we are going to relate the picture of each quadrant with the label of each quadrant. And then we score the abundance of each of the biological components. So for this abundance, we are using a brown blankets approach. The brown blanket is just a coverture abundance percentage survey. Here in the data sheet we are going to send you, you have, if you are not used to use brown blankets, or if you have not used it, is here it is like an easy guide. 0 is 0 for everything, and here we have like the scores that correspond to each percentage. We just go to the species level for the seagrass species. And for the green calcareous, the fleshy algae, the red calcareous, the stony corals, octocorals, and sponges, we just score like that, the big groups. This is going to help us then to analyze why the carbon storages on each site. Next. Okay, so here we developed this sediment texture rapid assessment. For me, like the hardest part to determine is the difference between sandy mud or muddy sand. Yeah, but okay, so here is the guide. If you take just a piece of sediment and you drop it in the column of water, if the sample forms a cloud and few particles sink, you have mud. If the sample forms cloud and less than half of the sediment you dropped sinks, is a sandy mud. And if the sample forms cloud and more than the half of the sediment you dropped sinks, is a muddy sand. I think the rest of the categories are like clear for almost everyone. Sand, coarse shell, I mean, is the sediment type is not hard to identify. When we have alimedas everywhere, rubble, live coral, or rock, or if you're sampling over rock, but I don't think that would be the case. This table, this rapid assessment for the sediment grain is also in the field guide you're going to have. And okay, we need to write down the type of sediment we are sampling in. Next.
So next, we'll just talk a little bit about the biomass course that we plan to take at every second quadrat. So what you see over here is actually a surveyor's flag placed where seagrass abundance was scored before. And these biomass cores, they're essentially, I think, 15 diameter PVC tubes. When we place it on the ground before taking the core, we have to make sure that the seagrass material that's inside the core is just seagrasses that originate from inside the core area. So seagrasses that just like lap into the core from outside, we have to take them out of the core before we push the core into the ground. Because we use this for above ground biomass, and we use the core diameter as our aerial extent, and so we don't want to have outside material in the sampling core. The core has a hole at the top, and before we retrieve the core, we close that hole to create a suction and make sure that we get all the sediment. When we take out the core, we use our hand to make sure that the root material doesn't fall out at the bottom, and then we transfer everything that's in the core above and below ground biomass into a mesh bag. Before transferring the material into the mesh bag, we remove that stopper, create that hole again on top of the core so all the material can fall out easily. And then we have all the seagrass material, a few algae pieces, probably a lot of sediment in this mesh bag. And by just shaking it a little bit in the water, we can remove a lot of that sediment because that's not anything we're interested here for the biomass course. We just care about the seagrass material as well as eventual algae material that might be there. So for this activity, we need biomass cores. Again, we will supply those to you and send them to you, as well as the mesh bags. And then we also need labels. I didn't mention that just now, but along with the material from the biomass core, we also want to put a label into the mesh bag that helps us identify from which quadrant the sample was taken. Generally, we do this to get estimates of above and below ground biomass for each seagrass species that is present. So these samples, after we took them, if we have a full field day, then we want to store them on ice and in the dark. If we have maybe a cooler with some ice cubes in there, just put the samples in there until you're at the lab. And in the laboratory, we have to process these samples. Because in the end, we are interested in the weight, and we are also interested in how many shoots there are per area. So we will generate not only above and below ground biomass in terms of weight per area, we will also get an idea of shoot density by counting shoots. So in the laboratory, our process is to prepare little aluminum tears. So it's basically a folded aluminum foil that we weigh before anything is in there. And we use that as a container to collect our seagrass sample. We then, for each biomass core separately, we separate out the seagrass material by species. And then we have another pile for all the non-seagrass material that is there. Then for each species, we count the number of shoots there are, write that down on our lab sheet. And then for each species, we separate out the life above ground from the below ground material. So leaves and shoots from roots and rhizomes. So they're all going to be in one piece. Of course, the leaves are attached to the roots and rhizomes. So we have to sever them with a razor blade, just cut them just where the shoot exits the rhizome. And then because we don't really want to measure the epiphytic material here, we're just interested in the seagrass material, we have to clean the seagrass leaves with razor blades, scrape the epiphytes off. So you just gently scrape a razor blade along the side of the leaf to remove anything that is growing on that leaf. And then once we're done with this, we can place all our species-specific seagrass above ground material in tears, or the below ground material in other tears. And we want to dry that material for roughly 2 days or so at 60 degrees before then weighing the dry weight. Now, these samples are not going to be lab processed for nutrients or so on. That's another set of samples. So you don't have to send any of this material across borders to us. So it's enough if you have the shoot count and then know the dry weight of above and below ground material by species.
Okay, we also, for the tissue nutrient and stable isotope analysis, we need you to collect at each site the seagrass material you find. For Thalassia, we need 7 shoots. For Syringodium, we need 30 shoots. For Allodule, we need 40 shoots. And if you find Allophila, we would need 50 shoots of these species. We also need you to collect specimen samples for herbarium, as Jim already told you. We want to have the sample like as complete as possible. That is with rhizomes, roots, with all the above ground material, so that we can preserve the material. As Jim told you, we are going to send you the press material, and we are expecting for you to return it back, to send it to the to the rest of the crew, of the CariCAS crew. Okay. Again, in the lab, each of the individuals we collected from each species, we need to scrape them with a razor blade gently to take off all the epiphytes. We just need to, we are using for this, we are using the above ground material, basically the leaves. So here is like the guide for the morphometrics. Only for Thalassia, we take the length and the width. And for the rest of for Allodule wrightii and Syringodium filiforme, we only measure the length. If we find Allophila decipiens, we count the leaves. And we collect all leaves of every species on the pre-weighed hairs, on the aluminum foil. We label it. We need you to dry each of the foils, each of the seagrass leaves in the oven for 48 hours at 60 degrees. And we need you to please record the dry weights. Then we need you to send this material back to the university to start running the analysis, the chemical analysis.
All right, I'm gonna just wrap up this in detail lab and field method discussion with the herbarium specimen. So here you collected a few fragments of each seagrass species present, which would include the rhizomes, the root, and then a few of the shoots. Now, we provide herbarium papers, and you would lay out the specimen flat onto these papers, make sure that the tissues don't overlap, and then top that with wax paper, put it in the herbarium press. Both wax paper and herbarium press is something we provide, and put a few sheets of absorbent paper, like newspaper, for example, in between, and then press the whole thing for a few days in a dry and cool place. And once these herbarium sheets are dry and pressed, you can use a pencil to label them. So you would write the latitude and longitude of where it was sampled, site name, which species it is, the depth, date, and then the collector's name. And that is it for this field guide. Now, I know there were a lot of questions in the chat, and perhaps that would be a good time now to respond to these and discuss all of these questions one by one. I see that Jim already answered a few of them in the chat, but perhaps would be helpful to reiterate some of them for everyone.
While people are gearing up questions and you're looking at them, we know that there are some countries in the Caribbean that have pretty much total bans of shipping biological materials out of their countries. And so sharing with us the herbarium specimens may not be possible, but we want to make sure we do everything following the proper laws and rules of the countries in which we're working. We'd still want you to collect herbarium specimens so that if it's necessary for someone to come in and visit, we can come in and visit. And Aldo is just asks, we have a manual that is in like alpha stage, and we're just getting rid of, we're just putting all the final pictures in it. So the manual will be a document that's available on the project webpage, Aldo. And all of this stuff is described in very close detail.
Thank you so much for that. I'm looking forward to reading it because I think that when we put in practice this, there is a learning curve, and it is very helpful for us if we have a guidance, like a written guidance for that.
We tried to make it step by step. In my lab, we've done this quite a bit. It's relatively easy for us to load up the boat, go out, do sampling of everything we want from a single site, and come back in the early afternoon. It's really it is a 1 day sampling per location, and if you're up at your site, you've got 2 locations. It should be about 2 days of field and maybe another half a day in the lab to clean everything up.
And if you don't mind, I have another question regarding the core analysis and the sample collection of the sediments with the core that you show. Mm-hmm. Sometimes here we have like seagrasses that are in muddy sediments. I mean, for those should be very easy to make the core penetrate into the sediments and take it out. They will remain in the tube. But there are some other cases where what you have is like carbonate sediments, and sometimes you find like a hard bottom platform just beneath of or below that tiny layer of sediments in which, and sometimes you don't know where you are stepping at. So what would be your recommendation? Because in that case, you will be sort of trying to collect the samples where you can, not where you should, or what happens if the site doesn't have like that kind of seagrasses where you have like a muddy basement for the community.
Sure. Although I've done this in many places around the world, and I've run into all of the problems you just mentioned plus more. One thing that we've done is we've made these core tubes out of stainless steel, and it takes a tremendous amount of force to break them. And what that allows us to do is sample in most back reef environments, but not all, because if there's broken coral fragments, say a staghorn coral fragment, these core tubes ought to go through it. But if you're running into large pieces that are larger than the diameter of the core tube, you won't be able to core there. And we seem to be able to core in sand quite well. And then the depth, I made a little comment about that earlier. I think you need to probe to know what the depth is, find what the average depth is, and then try to collect a core to at least that depth. This core is impractical if there's not at least about 15 centimeters or 20 centimeters of sediment. Okay. And if there isn't that much sediment, it is possible to just use the cutoff core tubes that we use for the subsamples directly through the surface and collect superficial samples. Most of the organic content in seagrass soils in the world is collected just of the superficial sample. So in this project, we're specifically trying to get that stuff that's deeper, where the deeper exists. Mm-hmm. But if you've got 30 centimeters and that's what your favorite seagrass meadow that you have all this work that you've done already, it's 30 centimeters deep, we'll take a 30 centimeter core. But I say it's going to work 95% of the time.
Thank you. Thank you so much for that, Jim.
There were a couple of questions in the chat regarding the amount of sediment cores and biomass cores. We talked a lot about different types of cores, so it's probably a little bit confusing. So just to reiterate, per seagrass meadow, there's only going to be one sediment core per site, and that's only one time. So it's not a repeated sampling through time. It's only this one deep sediment core at the center of the site. Now the biomass cores, those were 8 of them, so every second quadrat. And again, it's only this one time. So Ideally, you would go to the Seagrass Meadow once, take these cores, and then that's it for this project. You wouldn't have to come back. We are just going to provide one biomass core for you because you take a biomass core and then you empty the contents into a mesh bag, and then you can reuse the same core at the next quadrat that you take a biomass core. There were like a couple of questions relating to this. I just wanted to reiterate that factor. And Lindsay responded to a couple of questions in the chat regarding the materials. Yeah, again, as Jim just said, we have worked out this field guide that is providing the same information we presented here, but in a little bit more detail as well, illustrated. It's going to be available to everyone, as well as a recording of this workshop. So you can rewatch it. We also have a short video documentary of this process that is going to be available as a YouTube link we're going to share. So of course, we're always available for any questions. You can always reach out to any of the project management team. And so between these visual, audio and written documents, we hope that the methods are explained at some point. But also to reiterate a point that Jim made earlier, we are prepared to send field personnel from our lab to partner locations if you think that you would need help with the field and laboratory part of this. So Juan and I are ready to come and help with any of the sampling effort that is needed this summer.
Okay, here there is another question. I think Jim already answered, but it's related with the timelines of the project. So we are expecting to have the field samples by the end of the summer. It means the end of August. It gives us like enough time to process the samples in the lab, because by the end of December, we should have like the final report for the project. So if we could have all the samples from all sites by the end of August, we should be okay with these timelines we have. But again, it is just one field day to get each site. It's just one field day. On that day, the field day has two parts. One part is the coring. The coring will take you no longer than 2 hours. The second part is the scoring, the abundances. It won't take you more than 2 hours. So with half of a day in the field, let's say 6 hours, or maybe if you have to navigate too much, is 8 hours. With an 8-hour day in the field, you should have your samples done. That is for one site. So if you are planning to do 2 sites, it's 2 days in the field. Even though we, as Johannes mentioned it, we are planning, I mean, we have that in our plans to help, to bring any help you need. But yeah, we encourage you to send your samples before the end of August, if it is possible.
If you have put a question in the chat and you feel like it was not yet answered, I would like to encourage you to ask us now, raise hand, or just speak up, or maybe put it in the chat again if you feel like that. So I saw Savannah Berry ask whether by when we need to receive the dried or processed samples. Again, our ideal timeline would be to have these samples by the end of August. Yeah, we acknowledge that permitting process is something that's out of the power of most of us and rely on outside factors for that. But ideally, we hope that most of the partner sites can have the samples submitted to us by the end of August.
Here, Jorge is asking how many divers do we need? At least you need 2 divers. 2 divers is enough. 2 divers can do everything. 2 divers and a captain for the boat, but you need 2 persons in the water. That's all what you need. If you have a 3rd person, that would be useful, of course. There is always the need of an extra hand, because in the boat, the subsampling, for the subsampling, is good to have a 3rd person, but in the water, the minimum you need is 2 divers. If you could have 3, that would be great.
Yeah, let me add to that. Everybody has to follow their own diving rules and regulations. So part of this is if Curacao says you have to have 3, you have to have 3, but the work can be done at one location by 2 divers that are used to working with each other underwater.
So I see a question by Rodolfo Riojaneto, whether the cost for sending samples are covered by the project. Yes, so the packages that we send out with materials, they will include return labels already paid for and produced by us. So yes, those costs are covered.
And Aldo is correct. We want you to choose one site, hopefully that will become either is already part of your monitoring program or will become the focal point of future work. And then we would like you to work with us to collect another site so that we get enough sites from different kinds of environments to build a really good model to be used to interpret the remotely sensed data. And yeah, Jorge, we do need to move fast. A combination of COVID and then the complete collapse of being able to hire anybody because of the workforce problems after that have put us behind the 8 ball. We need to move it as fast as we can. And we get done what we get done. And I don't understand the true limitations of potential no-cost extensions from TNC, but that's a conversation I'll have with the TNC team. Right now, we need to get everything done by the end of this year.
We would send in the materials as soon as we schedule your field trip. If you're planning to go to the field next week, we will make sure that you will have the material before the scheduled date.
So sorry again, I am asking too much. So that means that every partner should sort of sign out the authorship agreement before you send the materials. Is that correct?
If you sign or don't sign, everyone needs to understand those expectations, and we will hold to those expectations. I don't think this is really a legal document, although other than an expression of what we expect. So the reason I'm hemming and hawing about this is I know a number of us won't know who will be the person that'll go into the field right now. So although you could potentially sign and whoever goes in the field with you may not, but then they'll be treated the same as you would.
Yeah, but what I'm saying is that you don't want to send materials to people who you don't know whether or not will be engaged or involved
That's true. We need solid expressions of interest saying we're all for this. We agree with what's been laid out in the authorship agreement, and we want to work for you in this project. As soon as we get that confirmation from you, we'll work to get all of the materials you need to you.
Perfect. That's great. What I'm gonna do is to sort of organize like an internal meeting with 3 partners, like key partners that we have here in the Dominican Republic. May I like transfer all the information that I got from this workshop, and from that point we can move forward, because my work here will be
Just dump this in the car. In the
Sorry, Mark, your microphone was open. Although this is not an exclusive club, we want everybody that wants to participate to participate. If we didn't send them an invitation, it was because we didn't know to, and you're welcome to share materials.
Okay, thank you so much, Mark. I will do so for sure. That's the way I see it, and I know that the group sees similarly.
Anyways, just one thing to add. We are like ready to have personal meetings with each of the teams, each of the collaborators, if needed. For the next weeks, I think our time is just to arrange and schedule these meetings with each one of you.
Mm-hmm.
And perhaps this is a good time to talk about what is going to happen just after this workshop. There are going to be a couple of more workshops for partners that couldn't make this meeting today. But once the workshop's concluded, beginning of next week, we will send out a Google form to everyone in which you can express your intent to participate in the CariCAS network. In the same form, we will also ask for potential sites that you think would be interesting to work at, as well as background, whether there's already data available from these sites, any other publications, knowledge, and so on. We will also, again, in this Google form, let you know what the materials are that we are sending you, what the materials are that ideally you have already. And we will ask you whether you will need field support. And lastly, what you think your timeline for fieldwork could be. And then as a next step, we wish to reach out really to every one of you and have one-on-one meetings with your people from your institution to hammer out details of what support you need, how we can provide it, and what's the best time for us to provide the support. So that we can schedule for our end, our support coming out to partner sites and helping you sample. But all of this will happen in the coming weeks. So individual site meetings are something we hope to conduct soon. And another thing that we will send out are the workshop certificates for everyone who took part. Once we know that you would like to take part of this project, we will send you the project material. And again, the recording of this workshop, as well as the field guide, will be shared with you shortly. So we understand we have to move fast. These timelines are relatively close. So we want to make sure that you have all the information you need to be able to judge when and how you can conduct this field work as soon as possible.
Yeah, and may I add to that, Johannes, that next month I will be at the AMLC meeting in St. Kitts for any of you who are planning on being there, and I'm giving a seminar about this very thing. So I'd be happy to set up a time that anybody that's going to be at AMLC would like to get together with me, and we can go through and put hands-on material and work out problems.
I'm just replying to another question in the chat. Jorge Herrera Severa. The site can absolutely be a coastal lagoon. Yes, that's possible. We do have sites here in Florida as well that we sampled already for this project. And one of them is a lagoonish place that is characterized more as an estuary rather than strictly marine. So yeah, absolutely. If seagrass grows there, we are interested in it.
All right, and so we've had everybody here for a long time, and I have to say that we've had over 40 people the entire time. Very few people have dropped off, and I'm going to take that to mean that a lot of people are interested and are trying to figure out how to be involved with the project. Please reach out to us if you need more information. We can have anybody else speak who would like to speak. And please just raise a hand if you would like to say something. Other than that, you've all been very generous with your time and so far today, and I hope that you'll continue to be very generous with your time to help us build this partnership and network out. Lindsay, you want to say for TNC?
I just want to thank everybody for participating. This has been really great meeting, and for the FIU team and putting it together. It's been really, really great.
All right, thanks all. Appreciate it. Look forward to hearing from you.